Alright, so this might go up before the actual Unit 3. This is the end of Unit 2 for us. It's our third, part of our third test is basically all of Unit 3 and then the last half of Unit 2. Alright, but we're going to start at genomics. So genome, entire complement of genetic information. So this is the genes, the regulatory sequences like promoters and stuff like that, and all the non-coding DNA. Genomics is the discipline of mapping, sequencing, analyzing, comparing genomes. I would wager that that name is going out of style in favor of bioinformatics, but we'll say genomics for now. More than 2,000 prokaryotic genomes have been sequenced or are in progress. The first, of course, was the RNA virus MS2, and then the other was Haemophilus influenza was the first eukaryotic genome. Or not eukaryotic, wow, I'm very off. The first cellular genome, which was in a prokaryote, which is Haemophilus influenza. And you can see by the date of 1995, that was only, that was 20 years ago now. I feel old. Yeah, but you can see that's a huge increase from 3,500, I guess that's the nearest round thing, then 1.8 million base pairs, so clearly advancing. And then now, of course, we've got human genome, which is 3 billion or whatever, so which can be done in like a day. So we're definitely way better at this now. That's just a bunch of prokaryotic genomes. Essentially, Craig Venter led the way with this, and the Human Genome Project, where they raced each other. And now we've got all the sequencing technology. So we know some things. For prokaryotic genomes, on average, it's 1,000 base pairs long, and there's 1,000 genes per megabase. And so you can imagine there's not a whole lot of wasted space in prokaryotic genome. If there's only a million, or there's 1,000 genes in a megabase, but there's often not more than a megabase of DNA inside a prokaryote. The upper limit at the bottom here is 12.3 that we know of. But roughly, the larger the genome, the more genes that are in it. Prokaryotic genomes have a huge variety of sizes. They can be very small, as small as viruses, to ones that are larger than some eukaryotes. Prokaryotes don't have the non-coding DNA present in eukaryotes, and but the largest ones are comparable to some eukaryotes. And genome size and complexity are not equivalent. They don't correlate. You can have the most genes is Trichomonas. Oh, it's right here, with 60,000 genes, and it's just a parasite, whereas humans are arguably more complex, especially when it comes to development. Usually when you hear complex, it's not referring to, oh, you can think in higher organisms in quotation marks. That's a really bad definition. What it really is how difficult the development is more, and how difficult maintenance of all the multicellular structures is more of what you mean when you say complexity. So next time you're asked, you should know. The smallest cellular genomes to date belong to parasitic or endosymbiotic parasites. So there's some obligate parasites that range from 490 kbp, so kilobase pairs, to 4,400 kilobase pairs. So that's relatively small. That's like, what, considering that last page, this means this holds like 500 genes at the most on the small end. And so those are obligate parasites, those things that basically can live on their own as long as they're inside the environment of a cell. Then endosymbionts, so these are things that only ever live within a cell, and essentially the cell has to take care of a bunch of functions for them. So it needs the environment of inside the cell. This means it literally is like part of the cell. Our mitochondria, we think, arose this way in some early eukaryote. 160 base pair genome. I've worked with plasmids that are smaller than that, so that's a ridiculously small genome. But for a full cell, 250 to 300 genes. So that's very small. I think this is supposed to say kilobase pair. I don't think 160 seems really low. Alright, and I talked about Trichomonas already. Alright, bioinformatics. Essentially lets you analyze, store, compare sequences and patterns and stuff. Complement of genes in a particular organism defines its biology, but the lifestyle is also important. Sequence similarity, comparative analysis, so you can predict things based on how like other things they are. And I believe the next slide, no, later on I'll get into why or what's the criteria for being close or not. Gene distribution in prokaryotes. Metabolic genes are the most. Replication and transcription are surprisingly a very small part of the genome. I assume that's because that's a very straightforward process. And then non-translated RNAs are in between those two. And that's just because so many basic RNAs are made out of them, like these tRNAs, which you need to make proteins, and then RNAs, which are part of ribosomes. So you also need those to make proteins. So it makes sense that they would be some of the most transcribed. This shows some various organisms, but the amount of chromosome space that's in a certain category. The thing I'd like to point out here is unknown is the biggest category of all of them. So it's basically over 30% of genes. Even in something as common as E. coli, we don't actually know what its genome is doing, what parts of its genome are doing. So that's really interesting to me, I think. I think know that metabolism is probably the biggest thing, and then I guess transport here is the next biggest. Yeah. Alright, microarrays in the transcriptome. Transcriptome is the mRNAs that are produced in a given set of conditions. So what's important to note is that unlike DNA, which is the same all the time, based on certain conditions and stuff you change, or prokaryotes as well change their mRNA makeup to try and respond. So if you're going to look at them, you need to study the condition under which you're curious about. Hybridization lets you see these, and okay, here's a DNA chip. You'll have 3 replicates of a gene. You get the basically cDNA of that. You affix it to the chip, and then you grow whatever it is, and then you isolate its RNA, and then you see if it binds, and and you'll have a probe that will fluoresce or something. Fluorescence, I think, is the standard now, and I don't know if there ever was a different standard. That's kind of misleading. Fluorescence is the way this works. That's what I'm going to say. And yeah, you can hybridize in conditions. I talked about that. And the advantage of this is you can do almost an entire genome and see what's going on, and it's based on these colors. Blue is cold spots, and then it's like a weather map where the red, or like thermal imaging, so the red is a hot spot, and yellow is a sort of hot spot, orange is a hotter spot, and red is the hottest, orange is the next, yellow is a little bit lower, green is lower, and then blue is nothing. What can you learn? So I have this little mnemonic here. Global condition, that's not right. Global conditions compared to identify unknown. So global gene expression and conditions, so the actual growth conditions. Compared, you can compare across organisms. Global conditions compared to identify, so you can actually identify an organism. Unknown, you can try and find out a function of an unknown gene. Proteomics, genome-wide study of the structure, function, regulation of an organisms' proteins. Pretty straightforward. This is what I was talking about with the bioinformatics. Greater than 50%, you have a pretty good idea of what a protein does. Greater than 70% is considered you have like 90 to 95% proof of what it does. The reason being that once you reach a certain critical mass of amino acids, there's only certain structures that the protein can hold. I'm assuming. Protein domains, these are distinct structural modules within a protein. And so you can look just for those domains. On the blast search, it's the conserved. CCD now, I think, is the tool that it's called. It's the conserved domains function. Very well stated. Alright, proteomics, a way to look at these is with a 2D polyacrylamide gel. And so this has two dimensions, a vertical and a horizontal. In the horizontal dimension, you're separating based on isoelectric point, better known as pH. And then in the vertical dimension, you are separating based on size. And so this is just a normal SDS page Western blot. And this is the 2D electrophoresis, which the only thing I'm not quite sure, I haven't actually run a 2D blot like this. I've run this kind of blot many a time. I'm just for the purposes of this, I'm assuming that you're generally supposed to assume that the basic side is the left and the acidic side is the right, but I have no idea if that's true. Metabolomics, complete set of metabolic intermediates and other small molecules present in an organism. So this is like glucose comes in, what are all the intermediates in the Krebs cycle and glycolysis and stuff? And MS is one of the ways that use this mass spectrometry. Okay, I would have said mass spectroscopy, but whatever, it doesn't really matter. Basically, you look to see disruptions in light to figure out stuff about these metabolites. And that's all we need to know about that. Oh, that moved the wrong way. Okay, mobile DNA, transposons and insertion sequences. So you can have horizontal gene transfer. So this is between organisms. That's not to produce a new organism. And the interesting thing for this is it may cross phylogenetic domain boundaries. So you may have, who knows, Salmonella enterica giving something to E. coli, which would be super bad. But anyways, directing horizontal gene flow, there's only, these genes are in distantly related species, so this is like you have E. coli and you suddenly find genes from Haemophilus influenza and you're like, hey, that doesn't match that. Or you look and they have a different codon bias for when they're doing translation, or the GC content is really far off, because GC content is important for stability in a lot of microbial genomes, so a difference would mean bad things. Right. Oh, and then species is usually based on polymerase similarity in the microbial world. You don't have the advantage as you do with multicellular eukaryotes like us in defining species. Alright, more gene transfer. Transposons may transfer DNA between organisms, which is different than you normally think of transposons, because normally it's within the same individual. And this is what this is talking about. You can get large cell chromosomal changes, so you can get multiple insertion sites. You can also get chromosomal rearrangements, which are the more... both of these are dangerous, but this one's probably more dangerous. Detecting uncultured, you can do a metagenome. So you just see the total gene content of everything inside an area. So there's been several large metagenome projects, and they found just this ridiculous diversity of stuff. Viral genomes. These are the most common genomes of all. They have a very high turnover rate. They also represent most of the genetic diversity on Earth, which is really interesting considering that a lot of people don't consider them life. I, however, am fascinated by viruses. Most virus genes are uncharacterized and show little or no sequence similarity to known genes. Yep, that's pretty much expected. Because we don't culture these, because they're mostly in seawater. Big surprise. Well, it was a big surprise for a lot of people, because we didn't realize the bacterial diversity in the oceans, which means we didn't realize the viral diversity in the oceans. Alright, sorry, I just had a deja vu moment. It was quite strange. Alright, Salmonella enterica. This is going to be a nasty little bug. It's gram-negative, it's paratrichous flagella. There's over 2,000 serovars, so this is different antibodies that could recognize the thing differently, the bacteria differently. So it's got a lot of diversity, and you can use the Ames test to find out if there's mutagenic chemicals for it. And there's varieties of this that cause bad things for us, so I think those will come up later. Information transfer in eukaryotes. This is just your basic. You cut out you transcribe your RNA, then you cut out your introns, then you put on a cap, a poly A tail, you transport to the cytoplasm, and then it goes to a ribosome to bind and translate. And you can have post-translational modifications after that. Alright, only 3% of eukaryotic DNA actually encodes for protein, whereas more than 90% of prokaryotic DNA codes. Eukaryotic microorganisms have fewer introns than higher eukaryotes, which makes sense. They're smaller, they need to use that space. We often have multiple copies of the same gene, which is quite strange, and we have multiple linear chromosomes. So we have redundancy. We also have telomeres and telomerase, so when this helps extend the end of our chromosomes, because otherwise we'd always lose, or we do actually lose a little every time our cells replicate, and that's thought to be involved in aging. But turns out if you turn this on more, you get cancer stuff happening, because that's tumor suppressors related to this pathway. So yeah. RNA processing, so you can have splicing, capping, and adding the poly A tail. I'm not going to dwell on this because this is not microbio. This is molecular bio, and it's not really my focus for this. Sorry to those who have not learned it before, but it is old hat to me. RNA polymerase II is the one that actually transcribes mRNAs. 3 does a bunch of specialty stuff, and then 1 is rRNAs, and this is the one that's highly conserved in like everything. So it's useful for bioinformatics, like I was talking about before. Transcription and translation, this is more of the same. Protein synthesis is much more complicated. Our ribosomes are larger. Let's see. 60 and a 40s as opposed to a 40 and a... Not 40, a 30 and a 50s. There we go. And let's see, there's more initiation factors in eukarya, which you would expect just because we have more protein diversity and more gene diversity. But most of these prokaryotes, eukaryotic mRNA is monocistronic, which is an important idea, is that there's only one gene per transcript, and then eukaryotic mRNA does not have a ribosome binding site. Instead, it has a cap. in prokaryotes, they have a specific sequence in the, what is that, the 5' UTR, that has the ribosome binding site sequence. So it's an interesting difference. Plasmids, genetic elements that replicate independently of the host. These are what get transferred in conjugation. They are not necessarily for survival, but often are helpful. This is how antibiotic resistance works. And they can be quite small, 1 kilobase pair to greater than 1 megabase pair. So actually they can get pretty big. But they're typically less than 5% of the bacterial chromosome. And how much an individual thing has is quite variable. This is just showing. Here's a plasmid. This is the normal DNA. Some general principles. Episomes can integrate into the host cell's genome, and this is similar to what's seen with prophages, and we'll talk about prophages a little bit. So just removing this is called curing. It's a cute name for it because you cure the thing of the plasmid. Conjugative plasmids are transferred via cell-to-cell. We'll talk about that more. And then, again, not essential, but often confer selective growth advantage. So it helps them outcompete their peers. Those are just some advantages, like Streptomyces produces streptomycin. And then virulent plasmids help them cause disease. Yersinia pestis has an injectosome plasmid, which counts as a virulence plasmid because it allows it to drill into cells. Alright, our plasmids, these are resistance plasmid, and these are the antibiotic resistant things. In several pathogenic bacteria, virulence characteristics are encoded by plasmid genes. And then bacteriocins, these are proteins produced by bacteria that inhibit or kill closely related species. So these are weird in that they kill off only things that are similar rather than things that are different, which is really counterintuitive, but it's one of those things where I guess, I don't know, keep your friends close, put your enemies closer. Just kill off all your friends. I don't know, this is some Game of Thrones logic here. Maybe it's because similar species are the most likely to try and take up the same ecological niche. That would make a lot of sense, actually. You just prevent them from using up resources that you need. Oh, this is a resistance plasmid picture. Before, when I talked about Salmonella, I was talking about Salmonella enterica, but the one that causes typhoid fever is Salmonella typhi, and it's got the same things, except it has the slightly different genome, and then it's got its own virulence plasmids. Alright, mutations, mutants. Mutations are heritable changes in DNA, but a mutant is any strain of a cell or virus that differs from the parental tame genotype. And then wild type is something isolated from nature. Mutations in an individual are usually deleterious, but overall in the population they're good for the population. And you want mutations to be inherited rather than just autosomal changes. Which in bacteria you're almost guaranteed to, since you just replicate the one genome, all your progeny are going to have whatever mutations you picked up. Whereas in multicellular organisms, that's quite a bit different. If I mutate a cell in my finger, it's not going to do much to my gametes. Alright, molecular basis of mutation. You can induce them. Some are, and that's with chemicals or radiation or something. Spontaneous, these just happen usually from free radicals, in particular oxygen.
or UV light. That's actually probably the more likely one. Point mutations. These change only one base pair. Often cause no change or just a single amino acid change. These are the various types of mutations that can result from just one base pair substitution. I'm not actually going to go through this because, again, not the focus of microbiology so much. Silent mutations don't do anything since one amino acid is changed. Nonsense mutation means you truncate the protein because you make a premature stop. And frame shift means the protein's out of frame, which can usually causes a truncated protein that doesn't make any sense when it's translated, and it folds wrong and just gets degraded. And you can lose gene function, which is relevant. Shifting reading frame, bad. Alright, so for most microorganisms with their DNA replication proofreading, 10 to the 6th to 10 to the 7th mistakes per kilobase. DNA viruses have error rates of 10 to the 3rd to 10 to the 4th. The mutation rate in RNA genomes is 1,000-fold more than in DNA genomes. The reason being, RNA-dependent RNA polymerases almost never have proofreading capabilities. And if they do, they're not as good. That's what this is trying to say. Shouldn't equivocate. Should just say it doesn't really have them. And this is why viruses can change so fast and avoid everything we throw at them. Mutagens, these are things that cause mutations. And there's nucleotide-based analogs. They look like nucleotides, but they're not. And they just get in the DNA and essentially mess with replication so that the wrong base gets put somewhere. Chemical mutagens, so these are like alkylating DNA, so it looks different. And then the acridines, these are intercalating agents that cause frame shift mutations. So nucleotide base analogs, they look like nucleotides and they put themselves in the DNA, and they're the wrong thing. You can have chemical modifications like alkylating, and then acridines, which are intercalating agents and cause frame shift mutations. This is just what some of them look like. Let's see. Non-ionizing versus ionizing radiation. Ionizing is the more severe. Ionized water to produce free radicals that mess with DNA. Non-ionizing, so this is UV. Pyrimidine dimers form. So these are thymine and cytosine that will form the thymine dimers, are the most common, and that causes problems. And you can use genetic engineering, but not the focus. Alright, Ames test. This is essentially you take a bacteria like Salmonella enterica, and you plate it on this plate, which you take the histine out of, and then before you've plated the cell, you essentially shoot up its ability to make histidine, and then you see these aren't growing very well, which means that they are not coping well with the lack of histidine, whereas these are growing very, very well, which means that somehow the broken gene they had has become fixed. And usually how that happens is that you put a chemical on these that you think might be a potential mutagen, and if it pokes the DNA enough, it'll re-enable this gene, is essentially. And that's going from oxytrophic to prototrophic. Prototrophic being wild type, like, or having an ability. Transformation. This is by which the process by which DNA is incorporated into a recipient cell, and it brings about genetic change. So some bacteria are just naturally transformable, and they'll pick up DNA from anywhere, and others you have to use something like heat shock or electroporation to get them to open up and take something in. Transduction. This is DNA derived from virtually any portion of the host genome and is packaged inside a mature virion. So this is a virus infects a cell, like a retrovirus usually, and then it tries to come out, but it brings some of the host DNA with it and produces an ineffective virus. And let's see. So in the lytic cycle, which, what does he call it? The virulent cycle. This little unsuccessful virus is a transducing particle because it's got part of the host in it. And then this is currently a transduced cell because it's a virion with host DNA. Not sure why that's in this chapter, but whatever. Conjugation. Bacterial conjugation. This is the mechanism of transferring plasmids. So you have a donor cell and a recipient cell, and you need a sex pilus for this. And essentially, the donor cell shoots its sex pilus at the recipient cell and then pumps across a plasmid. And the type of plasmid is the fertility plasmid. I suppose you could send a different kind, but this is the one that's got the capability already. It's about 100 kilobase pairs, so big enough to pack quite a bit. It contains genes that will regulate DNA replication, so it'll replicate itself basically, and it's got transposable elements that let it jump inside a host chromosome, and it'll have the Tra genes that encode the transfer function. So this is the pilus going from this onto the cell, and then it'll essentially reel this in until it's close enough to make this pilus a little bit bigger to let the DNA through. And this is showing it. Pilus pulled together, create some more bridge. Then one strand of the newly replicated plasmid spills over to the other cell and is replicated again in both. Transposable elements, discrete units of DNA that move from one location to another. They're in every domain of life. Can be anywhere from 1 in 1,000 to 1 in 10 million per generation. And Barbara McClintock, that's what she's famous for. Alright, this is Enterobacter rogenes, which is my unknown for my microbiome project, which is due Friday. This is an enteric bacteria, which you may have guessed by calling Enterobacter. Guts of animals, it's a coliform. It lives in the colon, that's what that means. And it's gram-negative, so it can cause disease, though not always. It's also found in dairy products. It's an opportunist infection, primarily involved in UTIs. You put it in media with lactose, and it is lactose fermenting. It has the lac operon, essentially. And then you would expect to be able to collect gas in germ tubes, and produces acid in its fermentation. It also is citrate producing, so it very, very definable organism. Major modes of regulation of gene expression. This is, you can turn it on and off genes as you need them. You can do it after the enzyme is made or before the enzyme is made. These are pretty straightforward. DNA binding proteins, mRNA, have a short life, so transcription typically requires that something binds to DNA to keep transcription going, and there's a lot of molecules that do this. Alright. There's sequence-specific matter, there's the one we talked about is helix, loop helix with the sequence-specific binding site in the major groove, often at a site of inverted repeats, and then the actual activator is on the other domain. Or stabilizing. Oh, not activity, stabilizing domain. This is true in the lac and trip repressors in E. coli. Alright, so it may cause something to catalyze a reaction, can block transcription, or it can activate transcription. Negative control or repression. This is where you just stop transcription until you need something usually. So this is arginine biosynthesis would be an example. I guess the lac chaperone is technically... not really. I mean, it is. If you say it in regards to beta-galactosidase. Alright, induction is the production of an enzyme in response to a signal. So, allolactose being present will induce cat to bind the cat binding site, and then the expression of beta-galactosidase. Alright. Hey, look, that's exactly what that slide just said. Yeah. Lactose, which will interconvert into allolactose readily, and that causes beta-galactosidase. The reason I'm skipping through this is because we'll talk about the actual lac chaperone in a little bit. Inducer is a substance that induces enzyme synthesis. Co-repressor is a substance that represses enzyme. Important, these are molecules acting on the actual regulator molecules, and collectively they're called effectors. The actual regulatory molecules are called allosteric regulatory molecules. So something that a repressor, one of these co-repressor molecules, binds to an allosteric repressor protein. And if it binds to the operator, it'll turn the gene off, which is sad, unless you don't need the gene. Operon is a cluster of genes arranged in linear fashion. Transcription is physically blocked when the repressor binds the operator. Induction can also be controlled by a repressor, so something can induce the repressor, or induce the system by removing the repressor from the site. As presence of glucose does. So co-repressor block causes the repressor to be on, inducer causes the repressor to fall off. Positive control, regulator protein activates the binding of RNA polymerase to DNA. And so maltose catabolism is an example of this. Maltose will bind to an allostatic activator, and that binds to the activator binding site. Or in the case of lac chaperone, it's called the cat binding site. Alright. And it's not part of the operator for real. Bars of positively controlled operants only weakly bind to RNA polymerase. Activated proteins help RNA polymerase recognize promoter. May cause a change in DNA structure, or may just interact directly with RNA polymerase to remove it from pause, probably. May be close or far away. So these are like enhancers in eukaryotic DNA. Lactose permease, it's an exchanger, or not an exchanger, it's a, what is this called? It's not really a pump. Oh well, it's not important. So this is the lac operon stuff. If you have allolactose, that's an inducer which causes the repressor protein to fall off. Or you can have glucose present, which will act as a co-repressor. You need an activator protein to get high expression of beta-lactosidase, and the inducer of that is cyclic AMP, because cyclic AMP goes up as you use up all your ATP, which means you've used up all your glucose. The cap site, and then cap binds cap. I talked about that already. And that's if there's lactose present, and really, this L-lactose is what's important to remove the repressor. If neither is present, you get low levels of theoretically, you'd get very low levels of transcription, and if neither is present, you get no transcription. If CAMP isn't present and but glucose is, you have the allosteric inhibitor there, also known as the repressor, bound as a repressor. Dioxic growth. It takes a little bit after you exhaust glucose to switch to using the alternative sugar source. Quorum sensing. This is an interesting little thing. Prokaryotes can respond to the presence of other cells that have the same species, and they can tell their population density, and this ensures that they have sufficient numbers to do things like release exotoxins. And in space, Salmonella's quorum sensing has changed and caused it to become more virulent. So the quorum sensing is by an autoinducer molecule that freely diffuses across the cell membrane, and then in a high enough concentration, it will trigger certain events. Acyl homoserine lactone was the first discovered. Doesn't really matter. One of the most interesting ones is the Lux operon in V. fischeri, which is responsible for bioluminescence in the ocean, which is super interesting. Why does it do it? Why does it have to wait till it has friends? I'm sure there's answers for those that are easy to look up. I just don't know them. Alright, and this just shows you if you have enough of these quorum sensing proteins, you'll get an activator protein. This will cause gene changes. These are quorum-specific proteins that are only produced when you have a quorum. Another thing is production of biofilms in Pseudomonas aeruginosa and virulence vectors of Staphylococcus aureus. Some microbial eukaryotes have it, and it's likely an archaea, though not proven apparently. Global control in the lac operon, and then so this is just a lot of different catabolic operons, like the lac operon, are affected by repression of various catabolites. And something like a flagella, you only need if you're not next to your resources already. Alright, and that'll be it. I know this one's, we did go through quite a few slides, but I know I skipped over some. This will be a shorter one just because the other one was quite a bit longer and it took me a while to do. If you have any questions, comments, or concerns, you can contact me at carebradio@gmail.com. That's carebradio@gmail.com. Until then, study hard. See you next time.